Our next speaker is Kleio Petratou, who's from the University of Bath. Thank you.
Thank you, and thank you to the organizers for this wonderful opportunity to be here today. This talk focuses on constructing gene regulatory networks that underlie fate specification of multipotent progenitors in the zebrafish neural crest. To begin, gene regulatory networks are defined as networks of genes whose interactions determine cellular phenotypes, and developmental processes are underlined by very highly dynamic and tightly regulated gene regulatory networks. To study these developmental networks, we use the neural crest as a model. So multipotent progenitors comprise the neural crest and are capable of giving rise to a very wide, astonishing array of derivatives, including peripheral nervous system components, bone and cartilage derivatives, and importantly for us, the chromatophore lineages of all vertebrates. So in zebrafish, we find three different types of pigment cell types, so the chromatophore lineages. We find black melanophores, light-reflecting iridophores, and the yellow xanthophores. And the reason that they are such great models for studying stem cell dynamics and gene regulatory networks is because they're very highly tractable, they're accessible, and easy to genetically manipulate. Moreover, they're deriving from a common progenitor. And currently in the group, we're in the process of characterizing the chromatoglioblast. This is a neural crest-derived tetrapotent progenitor that, undergoing partial fate restriction processes according to the progressive fate restriction model, can give rise to the different differentiated cell types, not only chromatophores, but also glia cells. Currently in zebrafish, only the bipotent cell melanoiridoblast has been characterized that derives after partial fate restriction process from this chromatoglioblast, and is able to give rise to both iridoblasts and melanoblasts. In this work, we're developing the gene regulatory network guiding this chromatoglioblast towards a mature iridophore fate. And to build gene regulatory networks, we use a systems biology approach. We start by screening for candidate genes of interest for our process and using the published literature. And we identify relationships experimentally between these genes to build a preliminary gene regulatory network, which we call Model A. We are able to mathematically describe this Model A using a set of equations and computationally simulate the network dynamics. So basically, we can tell that according to this network, how would gene expression dynamics would progress in our lineage. We are able to experimentally validate such predictions, and through a series of iterations of experimental validation and mathematical modeling, we derive a more sophisticated model that can better predict the observed dynamics. And this approach has been previously used in the group to characterize the melanocyte gene regulatory network. So the first step for the iridophore network was to identify candidate genes of interest. So SOX10, LTK, and TFEC were identified as mutants for these genes show lack of iridophores. MITFA mutants have ectopic iridophores, and finally PMP4A is a gene that very nicely is an early marker of the iridophore lineage. So TFEC expression, as shown by in situ hybridization, is a very good marker of the spatiotemporal development of the iridophore lineage. So in this gene, the expression of this gene marks progenitors of iridophores throughout their development, and finally the mature cell type. So at 19 hours post-fertilization, TFEC expression is found across the dorsal trunk domain of the zebrafish, and this domain corresponds to pre-migratory unspecified neural crest cells. We believe that a subset of these marked cells here are corresponding to the chromatoglioblast. 6 hours later, we find that the developmentally more progressed anterior trunk of the embryo is shutting down this pre-migratory unspecified expression in the neural crest. Instead, TFEC is shown marking the ventrally migrating specified progenitors, a subset of which correspond to melanoiridoblasts. At 30 hours post-fertilization, TFEC is expressed in a spotty pattern along the dorsal trunk domain of the embryo and also in ventrally migrating cells, and these cells now correspond to specified iridoblasts. And finally, at 48 hours post-fertilization, by live imaging of an embryo at this stage, we find that iridophores have a very distinct pattern, and this is exactly matched by TFSC expression. So to identify intergenic relationships between our genes of interest, we use both loss of function studies as well as some gain of function studies. We, for example, find that SOX10 is able to drive TFSC in iridophore development by looking at SOX10 mutants for TFSC expression. And what we find is that in wild type, those cells that I showed you earlier that are turning on TFSC expression and migrating are no longer present in SOX10 mutants. Moreover, we find that by overexpressing wild type SOX10, this gives rise to increased TFSC expression in a very early embryo, whereas overexpressing mutant SOX10 transcript cannot do that. Moreover, we find that LTK, which is a receptor tyrosine kinase, can bind to its ligand and upregulate indirectly TFSC expression. We see that by looking at LTK mutants for TFSC expression and show that these iridoblasts that I showed you earlier are now not marked by TFSC. And the only remaining cells here have very weak TFSC expression. And similarly, TFSC, we find, also drives LTK to maintain the gene's expression in this lineage. And we show that by knocking down and also knocking out TFSC in other experiments, and we find that LTK is significantly decreased. So, building on such a data set, we're able to derive a preliminary gene regulatory network that drives iridophore development. We call this model A. Here, solid arrows show proven direct interactions, whereas dashed arrows are not yet known whether direct or indirect in most cases. We're able to characterize this network and describe it mathematically using a system of ordinary differential equations. And I'm not going to go into those equations, but by numerically solving them using software MATLAB, we're able to derive the predictions as to how the concentration of the protein product of each gene can progress over time in the iridophore lineage according to this model. So what we found is that in the wild-type context, PNP4A, LTK, and TFSC stay upregulated at late stages of development. And this is a biologically accurate result because all these three genes are late iridophore markers. Moreover, we find that SOX10 is, well, we've actually defined SOX10 to be downregulated according to our knowledge at this point from the literature. And we find that MITFA shows a rise and then drop later on development. And we validate this result for MITFA using double fluorescent expression studies. So we double label for MITF and TFSC transcript using RNA-scope protocol. And what we find that is at 24 hours post-fertilization, TFSC positive iridoblast specified cells are also expressing MITFA, whereas at 48 hours post-fertilization, these two genes are expressed in distinct cell populations, therefore justifying a rise and then drop of the MITFA in the iridophore lineage. Moreover, looking at predicting the dynamics when simulating an LTK loss of function, we find that these qualitatively are biologically accurate. So what we see here in the prediction is that PNP4A first rises and then drops, and TFSC is doing the same in an LTK mutant. So by looking at LTK mutants with our experiments, we find that this is indeed true, and we have a gradual decline of both TFSC and PNP4A expression at late stages. And looking at the SOX10 mutant simulation of loss of function for SOX10, we find that TFSC, LTK, and PNP4A are predicted to stay upregulated throughout development. And we find that looking at the SOX10 mutant, this is indeed true. So TFSC and LTK are both staying upregulated in pre-migratory cells. However, PNP4A is eliminated throughout development. And therefore, we derive a model B, according to which SOX10 is absolutely necessary for driving PNP4A expression in development. And the predictions of this model now are both that LTK mutant and SOX10 mutant simulations look biologically accurate. So here we find that PNP4A always stays downregulated. However, in the wild type, PNP4A is later turning off. And this is a direct consequence of SOX10 being defined from the literature also turning off. However, this is a biologically inaccurate prediction because PNP4A is a late iridophore marker. And so we look at SOX10 expression and how these dynamics are progressing in iridophore development. And we found that by co-expression studies, LTK positive cells always express SOX10 throughout development. So 100% of these cells will express SOX10. And moreover, SOX10, as shown by in situ hybridization, the expression is always coinciding with mature iridophores. And therefore, we derive model C, according to which TFEC upregulates SOX10 in an iridophore-specific way. And as predicted by this model, in the wild type, PMP4A stays upregulated. However, MITFA is doing the same, being a direct target of SOX10. That makes sense. However, MITFA is a melanocyte-specific gene and therefore should not be upregulated in the iridophore lineage. Therefore, we're trying to look for a repressor of MITFA in the iridophores, and we suggest model D, according to which TFEC drives an unknown factor R that represses MITFA in this lineage. And trying to look for factor R, we first test FOXT3 being a very good candidate based on the currently published literature, but we find that this is not actually a good candidate for our network because only 50% of LTK positive cells actually express FOXT3 and not 100% that we're expecting to see. And moreover, MITFA positive cells are reduced in FOXT3 mutants, which also does not coincide with FOXT3 repressing MITFA. And to conclude, we develop, using an integration of mathematical modeling and experimentation, Model A for iridophore lineage towards a more sophisticated Model D. Future directions for this work include identification of direct versus indirect interactions using chromatin immunoprecipitation and sequencing. We also want to quantitate gene expression and be able to better predict gene expression dynamics. And finally, we integrate Model D with a melanocyte-established gene regulatory network to build the melanoiridoblast fate switch network and better understand how the neural crest becomes specified. And I would like to acknowledge the help of both my supervisors and colleagues, especially Tatiana Sabhankulova for the overexpression studies, our collaborators, and the organizations that helped me being here today. And thank you very much.
I guess there's one way of accomplishing SOX10 expression, which would be to have the same enhancers always use SOX10. But is it also possible that there's iridophore-specific enhancers for SOX10? So as you start evaluating your regulatory element, like it switches an element.
Right, so in terms of how SOX10 becomes upregulated. So we define it as being upregulated by TFEC because TFEC is currently being believed to be the master regulator of the iridophore development, similarly to MITF being such. And we are currently using CRISPR mutations for TFEC in the process of validating that prediction. So it's an experimentally validatable prediction. So there could be other ways of doing that, but we can disprove this model that is currently making sense to be this way and move on to other predictions. But this is a very good this is a very good idea that we can test. Thank you. Thank you.
So that concludes the gene regulation and RNA biology session for today. I'd like to congratulate the speakers for not only keeping to time, but for their really interesting talk. So thanks very much, everyone.